Good morning, all of you. Today's topic is structure and functions of the plasma membrane. First of all, structure of plasma membrane. There are three main parts of the cell. First, plasma membrane, second, cytoplasm, and third, nucleus. Now, structure of plasma membrane. The basic structure of plasma membrane is the lipid bilayer, two back-to-back layer, and it is made up of three types of lipid molecule: phospholipids, cholesterol, and glycolipid. About 75% of membrane lipid are phospholipids and it's contain phosphorus. 20% are cholesterol and it's contain steroid with OH groups, and about 5% are glycolipids and it's contain carbohydrate with lipids. The bilayer arrangements occurs because the lipids are amphipathic molecules, and amphipathic means it has both group polar and non-polar parts. It is diagrams of cell membrane. Now we will discuss first of all first lipid. It is phospholipid. The phospholipids contain, in last slide we have said that lipid is amphipathic molecule, and amphipathic means it's contain both polar and non-polar parts. So polar parts is the phosphate containing head and which is hydrophilic. In phospholipid, polar part is phosphate containing head. This is phosphate containing head, and it is hydrophilic groups of phospholipids. Second, non-polar parts of phospholipids are two long fatty acids tail. This is two long fatty acid tail, and which are hydrophobic, means one hydrophilic and another one is hydrophobic parts. Hydrophobic hydrocarbon chain. The phospholipid molecule in the bilayer with their hydrophilic heads facing outward. Always it is these hydrophilic heads remain always outsides, and fatty acid tails remain inner sides. In this way, the head face always watery fluids on either sides, cytosols on insides and extracellular fluid on outsides. Hydrophobic fatty acid tails in each half of the bilayer point. These all are hydrophobic fatty acids. Hydrophobic fatty acid tails in each half of the bilayer point toward the one another, and its form one non-polar hydrophobic regions in membrane interior part. Now it is second lipids of plasma membrane, and that is cholesterol. Cholesterol molecule are weakly amphipathic, and it is interspreads among the other lipids in both layer of the membrane, means in upper sides, lower sides, in both layer, and it is generally interspreads, means it is present in other lipids. The tiny OH groups is only polar regions of the cholesterols, and it's form hydrogen bonding with polar head of phospholipids and glycolipids. And the steroidal rings and hydrocarbon chains of cholesterols are a non-polar part, means here in cholesterol OH groups it is polar part, and steroidal rings it is non-polar parts. Polar parts make hydrogen bonding with polar parts of other lipids, and non-polar parts are also combined fit with other non-polar parts of other lipids. Third lipid is glycolipids. The carbohydrate groups of glycolipids form a polar head, and their fatty acid tails are non-polar. Its name indicates glyco means it is carbohydrates and lipids. So glycolipid is a combination of carbohydrate and lipid. Now the bilayer are asymmetrics. Bilayer of plasma membrane are asymmetric. Why it is known as asymmetric? Because glycolipid appear only in the membrane layer that face the extracellular fluids. It is not, glycolipid is not present on the both layer, but it is only present on those membranes which are faces extracellular fluids, which one reasons That's okay, two sides of bilayer are asymmetric or different. So from these points we can say that bilayer of phospholipids are asymmetric, means both are not similar. Now, second molecule of the plasma membrane structures is membrane proteins. Generally, there are two types of membrane proteins present in the plasma membrane. One is integral and second one is peripheral, according to whether they are firmly embedded in the membrane. First, integral protein that is extended into or throughout the lipid bilayer among the fatty acid tails, and it is firmly embedded in it. In these figures, we can see these are integral proteins which are extended into or throughout the lipid bilayer among the fatty acid tails and are firmly embedded. Like membrane lipids, integral membrane proteins are amphiphatics. These proteins are also amphiphatics, means it has both groups, polar and non-polar. Their hydrophilic regions protrude into either the watery extracellular fluids or cytosols, and their hydrophobic region extend among the fatty acid tail. As their name implies, peripheral proteins, now second protein is peripheral, and peripheral proteins are not firmly embedded. Integral proteins are firmly embedded, peripheral proteins are not firmly embedded, but they are associated more loosely with polar head of membrane lipids or with integral protein at the inner or outer surface of the membrane. They are loosely bound with some part of the lipids and also with the integral protein. Next protein is glycoproteins. Like glycolipid, glycoprotein is also combination of carbohydrate and proteins. Many membrane proteins are glycoproteins, means proteins with carbohydrate groups attached to the ends that protrude into the extracellular fluids. And in glycoproteins, the carbohydrates are generally oligosaccharides. Oligosaccharides means its chains contain 2 to 16 monosaccharides, and they may be straight or branched. Carbohydrate portion of the glycolipids and glycoproteins form an extensive sugary coat, and it is known as Glycocalyx. What is Glycocalyx? One thin carbohydrate layer present, extensively present sugary coat on the cell membrane. It is known as Glycocalyx. The pattern of carbohydrate in glycocalyx varies from one cell to another cell. It is different from cell to cell. So, glycocalyx are known as signature of the cell. For example, for white blood cells, ability to detect the foreign glycocalyx is one basis of the immune response, and that help us destroying foreign particles or invading microorganisms. Now, second portion of today's lecture is function of the cell membrane, function of the membrane proteins. So first function is some membrane proteins form ion channel. That is first function. It form ion channel, pores and holes, and through these specific ions such as potassium ions can flow to get into or out of the cell, and most of ion channel are selective and they allow only single type of ion to pass through. Okay, the first function is it forms channel, pore, hole, and through this channel some specific ion can easily pass or can out from the cell. Second, other integral protein act as a carrier. Second function is membrane protein act as a carrier, selectively moving a polar substance or ion from one side of the membrane to other, and its carrier are also known as transporter. Now, third function is integral protein called receptor and act as a cellular recognition sites. Each types of receptor recognize and binds a specific types of molecules. For example, insulin receptor binds the hormone insulin, and a specific molecules that bind to a specific receptor, and it is called the ligand of that receptor. So here some integral protein that work as act as a receptor and some specific molecule bind with the receptors, and after binding the receptor it can produce its effect. Some integral proteins are act as enzymes, and we all know that what is the functions of enzymes. The enzymes that catalyze the specific chemical reaction. So here integral proteins are enzymes, and so it's catalyze specific chemical reaction at the inside or outside the surface of the cell. Integral protein may also serve as a linkers, and which anchor proteins in plasma membranes of the neighboring cells to one another, or to protein filaments inside and outside the cells. And so peripheral proteins also serve as enzymes and linkers. Last function is membrane glycoproteins and glycolipids often act as a cell identity markers. They may enable a cell to recognize other cell of the same kind of tissue formation, or to recognize and respond potentially dangerous foreign cells. Now, one example, the ABO blood types markers. When we receive a blood transfusion, at that time the blood type must be compatible with each other. Thank you very much.